All right, today we're pulling back the curtain and going inside some of the most secure, most important scientific spaces on the planet. We're going to demystify this thing called biosafety and get a peek inside these incredible labs. Let's get right into it.
So let's start with a big question, right? If your job is to study the world's most dangerous germs, we're talking bacteria, viruses, all of it, how on earth do you do that without getting sick yourself, or worse, letting it out into the world? It's a really high-stakes puzzle, and it's one that science has gotten very, very good at solving.
You know, for thousands of scientists out there, handling stuff like this is just a normal Tuesday. This isn't some sci-fi movie scene, it's the daily critical work that leads to new medicines and vaccines. And to pull it off safely, they depend on a really powerful and systematic set of rules. And that whole system, that whole set of rules, it's called biosafety. So what exactly is it? And maybe more importantly, why does it matter so much to every single one of us? Well, at its heart, biosafety is all about one thing: containment. It's a whole collection of practices and gear designed to keep the scientists safe and to keep the public safe. And look, this isn't just theoretical. There have been rare but really serious lab accidents in the past, and that's why these rules are basically written in stone. It's all about creating a protective bubble, making absolutely sure that the dangerous stuff stays right where it's supposed to be, inside the lab. So how do they actually create that bubble? Well, it pretty much boils down to two fundamental types of barriers. You can think of them as your first and your second line of defense against anything nasty. On the one hand, you've got what's called a primary barrier. That's your personal protective equipment, or PPE. You know, the stuff you actually wear. Gloves, lab coats, masks, sometimes even a full bodysuit. It's what physically separates you from the microbe. And then you've got the secondary barrier, and that's the lab itself. The building, the special air filtration systems, the locked doors, it's the whole environment, engineered for safety. Okay, now this table is basically our roadmap for the whole system. It shows how everything scales up. Depending on how risky a germ is, the rules and the equipment get more and more intense, climbing through 4 different biosafety levels, or BSLs. So let's use this as our guide and level up, starting from the bottom. We're going to start our climb with biosafety levels 1 and 2. These 2 levels are really the foundation for almost all of the biological research that happens anywhere in the world. So, BSL 1, that's your starting point. Think about your high school or college science lab. You're working with microbes that aren't really known to make healthy people sick. But BSL 2, that's a real step up. This is the workhorse level for most clinical and research labs, where they're handling things that pose a moderate risk, like the staph bacteria or hepatitis viruses. So what's the big difference at BSL 2? Well, you've got the standard stuff, of course, like lab coats and those biohazard signs we've all seen. But the real game changer is that piece of equipment called a biological safety cabinet. It's basically a special ventilated hood that you work inside of anytime you're doing something that could kick up tiny infectious droplets into the air, droplets you could breathe in. It keeps both the scientist and the experiment safe. Okay, now we're taking a big leap. Welcome to level 3. This is where you're dealing with microbes that are seriously bad news if you inhale them. We're talking about things like the bacteria that causes tuberculosis or other dangerous viruses that can cause lethal infections. Yeah, a BSL-3 lab is totally designed for next-level containment. Every single bit of work with the infectious agent has to be done inside one of those biological safety cabinets, and the lab itself is usually in a totally separate, restricted area of the building just to add another layer of security. So, you take everything you do in a BSL-2 lab, and then you add some major upgrades. Access is super restricted, but the absolute coolest, most important feature is the directional airflow. The entire lab is kept under negative air pressure. That means air from the hallway always flows into the lab and never out. It's like a one-way street for air, making sure nothing dangerous can ever just drift out. It's pretty brilliant, actually. And here we are, the top of the mountain, level 4. This is the highest level of biocontainment there is. There are only a handful of these labs in the entire world, and they're reserved for the deadliest pathogens known to humanity, things like Ebola and Marburg virus, where there's no vaccine and no cure. Just look at this picture. This is the BSL-4 world. These scientists are working in what people call spacesuits. They are completely sealed off from their environment, breathing clean, filtered air that's pumped directly into the suit. This is the absolute peak of personal protection. To work in one of these labs, you need everything from BSL-3 plus procedures that are, no joke, Straight out of a movie. You go in through a series of airlocks, you leave through a chemical shower to decontaminate your suit. All waste is basically superheated in a giant pressure cooker called an autoclave before it leaves. And all your work is done either in a totally sealed box or in one of those positive pressure suits. The suits are incredible. Air is constantly pushing out of them, so even if you get a tiny tear, the air pressure keeps any germs from getting in. Okay, so we've climbed all 4 levels. We've seen the spacesuits, the airlocks, which really begs the question, why? Why go through all of this incredible trouble and expense? And the answer really couldn't be more important. This whole system, it's the absolute foundation of modern public health. It's what allows scientists to safely study new and emerging diseases, to develop the vaccines that save millions of lives, and to be ready to respond when a new outbreak happens. It keeps the researchers on the front lines safe, and by doing that, it keeps every single one of us safe too. Which, of course, leaves us with one last big question to think about. We know science never stands still, right? New viruses pop up, our technology gets better and better. So the question we should all be asking is, how will these incredibly important safety levels need to change and adapt to keep us all safe from the challenges of tomorrow?